Thank you. I have no idea what happens next. I guess I get asked some questions. I can make a brief statement. Well, I haven't prepared anything, but that hasn't usually stopped me, so I'll try. Yeah. Okay. Actually, I didn't receive the phone call from the Academy. I slept through it. What happened, through a mistake a couple of days ago, I inadvertently adjusted our phone such that it had a very soft ring. So I woke up this morning at 10 after 6, and I realized that there was a phone ringing somewhere, and I thought it was somebody else's apartment, and I was a little bit annoyed that they weren't answering their phone. I then realized, because it was after 6, that they must have announced the Nobel Prize in chemistry, and so I decided to find out who the schnook was that won it this year. So I opened up my laptop and found out that I was the schnook. The other two people are very good scientists. So it was quite a surprise to see that, and I woke up my wife and I said, "Hey, look at this! This is very amusing." So I didn't do that, and then the phone started ringing a lot more. So I actually missed the call from the Swedish Academy, but then talked with them later.
And your discovery was made 14 years ago?
Actually, the discovery was made about 16 years ago, but we published it 14 years ago in February. There's a lot of good science around that people get recognized for, and we were just fortunate enough that this year was the year that Osamu Shimomura, myself, and Roger Chen were recognized for GFP. If what you're asking is, what's the usefulness of this? Why is this protein something that very many scientists use? That I can easily answer. But I have to give you a little bit of history. So the history is that when people have always been interested in how cell components work and how they interact with each other. And people have for many years wanted to know where particular cell components are, particularly proteins, are in cells. And they've been able to localize them, to look at where they are in cells, but usually in preparations that are fixed, that is, dead tissue, and you say, where is it in this dead tissue? The wonderful thing about green fluorescent protein is that we can engineer cells so that they make the green fluorescent protein, either by using the control sequences that turn genes on, and so we can ask the question, in what cells is the gene turned on? Or we can ask the question, as was first done by Tula Hazelrig, who's back there, here, of asking what happens if we put GFP onto a protein? Can we actually see where the protein is in a cell and how it might move from one cell to another or from one part of the cell to another? And we can do this all in a living organism. And the reason is that all the previous methods, you had to get stuff into the cell, either to cause an enzymatic reaction or to get the antibody in to bind to the protein. Here we're having the cell make the component. And the only thing you have to add is light. And because these proteins are fluorescent, you shine light of one wavelength on the specimen, and you get light of another wavelength out. Very much like a black light in the 1970s disco, that you're able to see this. But that serves as a lantern for which you can now follow where the protein goes or what cell is expressed. And you can actually ask a number of very nice questions and look at cells and animals in real time. So for example, if you're interested in trying to understand the basis of infection, and you want to know what happens when a organism, let's say a mouse, is infected by a bacterium or infected by a virus, you can make it so that the bacterium is labeled by GFP, or the virus will make a protein that is labeled with GFP, and we can follow the infection. Now, we don't do that in my laboratory, but other people have. Other people have asked about questions of parasites and their hosts. How do the parasites interact with the host? By labeling the parasite and looking at the infection of the parasite. And another aspect is to study cancer. If you're interested in how do cells metastasize, how does a cancer spread through the body, you need a way of looking at the cancer cells. So by labeling the cells with GFP and looking at this in a mouse, it's an opportunity to follow the development of a disease. Now, in addition, there's a lot of very basic research designed at trying to understand development in general and how genes work and where they're working. So this has become a very popular tool in cell biology and in developmental biology. And it's through the discovery of these fundamental processes of development and cell interaction that we learn the biology that's really necessary for an understanding of human disease. So we don't really study human disease. We study the basic biology, and that's what's very important. This research, which was funded by NIH, is all research that is directed not towards solving a particular problem, but is developing a tool that we can then use in a wide variety of problems. And that's what the importance of it is.